My name is Dan Bunker. Thank you for joining us today. This is Rebecca Panko's dissertation defense, and it's been my great pleasure and honor to be her dissertation advisor. Before we start, I want to talk about the protocol, the plan that we have in place for this virtual defense here, because I believe this is perhaps our first attempt to do this all on WebEx. So first of all, please keep your camera off unless you're asking a question, and also keep an eye on your mic and make sure that's generally off. At the bottom of your screen, you'll find the chat widget, which you can click on, and that will allow you to, you can just type if you have a question at the end, just type in question, and we'll work through the questions in the order that they're posted. In a minute, I'll introduce the Dissertation Committee, and then I'll introduce Rebecca, and Rebecca's presentation should last roughly an hour, more or less. And then at the end of the presentation, we're going to ask that you all unmute yourselves, and so that we can all have a rousing round of applause, and then mute yourself afterwards, if you would. After that, the committee chair, who's Klaus Holzapfel, will moderate questions from the audience. So again, you can just type question into the chat widget, and we'll work through them. As with face-to-face defenses, the Dissertation Committee will reserve their questions for the closed portion of the defense afterwards. After the question and answer period, we'll unmute for another round of applause, and then the chair will ask everyone to leave except for the committee and the candidate. So with that, I'd like to introduce Rebecca's Dissertation Committee. So committee members, if you would turn on your video for a moment, I'd like to introduce you. Dr. Jessica Ware joins us from the American Museum of Natural History. Dr. Phil Barden joins us from NJIT. Dr. Emily Josephs is Rebecca's external member and joins us from Michigan State University. And finally, the chair of the committee is Dr. Klaus Holzapfel from Rutgers, Newark. Super. Thank you very much for participating and for advising Rebecca throughout her dissertation. Super valuable. Next, I'd like to introduce Rebecca Panko. So Rebecca earned her Bachelor of Science in 2014 from the City College of New York, where she worked with Dr. Amy Burkhoff, who I believe is in attendance today. Mm-hmm. As part of her undergraduate work, she studied whether compost leachate could be used to water plants in an artificial salt marsh. And her first publication was in 2015 that resulted from an REU investigation looking at interactions between invasive herbivores and their long-term impact on New England hemlock forests. Rebecca has served as a teaching assistant at Rutgers Newark and at NJIT. And in particular, she's been the instructor of record for Plant Kingdom at Rutgers since 2018. So she's been the professor for that course, which is a service in teaching above and beyond what most grad students have the opportunity to do. She's also contributed service to the department. In particular, she served as the graduate student representative to the biology graduate faculty as a member of the biology student leadership. She's also served as our biology rep to the Graduate Student Government Association. So it's been really a wonderful thing to be able to work with Rebecca. She's smart, she's enthusiastic, she's motivated and driven, and just developing into a really fantastic scientist, and it's been a real joy. When Rebecca first approached me about joining my lab, she told me that she wanted to work on urban ecology and urban evolution, and I was super excited about that because when I first came to NJIT, it seemed obvious to me that as an ecologist and evolutionary ecologist, I should be working on urban systems. And so with Rebecca, I've been able to do a lot of that, and it's been very exciting. The system that Rebecca decided to work in involves this cute little weed called Capsella that grows in cities all over the world. And Rebecca really should be credited for developing this system really from scratch. Other people have researched this plant, but Rebecca has taken the lead in developing this research system to ask these questions about urban evolution. And with that, I'll introduce her and her dissertation, which is entitled Plant Evolution and Urbanization, Quantifying the Effects of Natural Selection in Shaping Shepherd's Purse, Capsella pastoris populations in New York City. And with that, Rebecca.
Thank you, Dan. Oh man, thank you guys. What, Dan?
Oh, I have to pass the
Oh yeah, I can't
little
share yet.
little baton to you.
Well, while you do that, I'll just say thank you. So thank you so much, Dan, for the lovely introduction. Thank you all for joining me here today. This is a very surreal experience. I never ever thought that I would be defending my PhD in my home office in slippers. So it's just been a whirlwind since this whole thing started. And we, and the school shut down and things like that. And I have had to like move things to my house, my lab to my house. And it's just been, like I said, a whirlwind, but I am beyond excited to share my work with you guys today. And I am just so happy to be at this point. Yeah. So just gonna soak it in for a moment because I've been waiting for this for a long time. All right, so let me pull up my slides and give me 1 second there. Okay. I will warn you now that I live on the first floor of a Brooklyn apartment, and so the front door is located very close to this wall. And so there might be a little incidental noise of people going in and out or of my radiator turning on. I hope not, but just an FYI. All right. Can everyone see the slide? I think you can. All right, so I'm going to get started. Thanks again. So yes, as Dan mentioned, my dissertation topic is plant evolution and urbanization, quantifying the effects of natural selection in shaping shepherd's purse or Capsella bursopastors populations in New York City. So urbanization is increasing worldwide. This is a worldwide phenomenon. And when urbanization gets built, but also spreads, it creates a lot of dramatic changes to the land. So as you can see, these are some examples of some really, really big cities, right? You have Beijing on the top left, Mexico City on the top right, New York City on the bottom left, and then Istanbul on the bottom right. And just with your eyes, if you're looking at these aerial shots, it's very, very clear how transformed the landscape is. And part of the reason that urbanization is increasing worldwide is because of the amount of humans that are moving to and living in cities. And this is causing existing cities to expand, but it's also creating new cities in areas where there currently wasn't such a built infrastructure. So in 2009 was the first time that the global population of humans in cities outnumbered the global population of humans living in rural areas. And this trend has continued since then, and it's expected to continue continuing for the foreseeable future. And so you can see here, if things, we've already gone almost like from the 1950s, the number of urban residents has increased almost fourfold, right? So it's quite a large number of humans. Let's look at what this kind of looks like on a map of the world, right? So this is data from the UN that's been visualized by this company that makes pretty maps. And basically, you can see here, this is in 1950, and there are some areas that are already becoming urbanized, but not that much. And then if you go to 2020, what you can see here is not only did areas, so let's go back for 1 second, not only did areas that were already urbanized in the 1950s grow in human population, but look at how many new areas have been urbanized since in that 70-year time, right? So this is what we're talking about with the increase in it worldwide. And if you've ever been in a big city, cities can be kind of tough environments to live in. So if you're a plant living in a city, this might include different types of things, right? Maybe you have to live in a grate growing through the sidewalk. Maybe somebody comes around and ties a bunch of toys onto you, and then it gets rained on and it's hundreds of pounds of weight on your branches. Or maybe somebody comes by and graffitis you or pours concrete in your street tree pit. So it's like, oh man, so many different things that can happen, so many different factors that can impact plants in cities. So many. So some of the things that come about via urbanization are some very well understood syndromes of environmental change. So for example, habitat fragmentation. If you look at this top left image, this is an aerial shot of Central Park from New York City. And as you can see, it's a very clear distinction of this fragmented green space surrounded by this hardscape, right? So that's an example of fragmentation. You also have habitat degradation, which is when things occur that cause the land to be in a state that's almost no longer inhabitable to certain organisms. Another thing that we see in cities is an increase in altered abiotic and biotic stress factors. So first we'll just talk about the abiotic factors. In cities, you tend to have higher relative air temperatures. You tend to have more water stress in cities. You tend to have higher levels of salinity in the soil if you're in a city that regularly applies de-icing salt in the wintertime. Of course, more pollution in cities. And then even though today we're going to focus on the abiotic things, we have to also mention that there are also biotic factors that are changed because of cities. So part of the reason that cities have these changed abiotic factors is because cities have high proportion impervious surfaces. Impervious surfaces are surfaces that water cannot infiltrate. So if it rains, the water can't be absorbed by the soil. So this is streets, sidewalks, buildings, things like that. And this is a map, this is from Google Maps of our area, of our region. And again, even with the naked eye, you can see the differences in the landscape. But if we were to put a land cover map on top of this that highlights impervious surface in red, it becomes very clear how much more impervious surface there is toward the urban core. When you have an increase in impervious surface, you have different changes that can occur. So one of these changes is an increase in drought and an increase in flooding events. We will talk more about that going forward, but as you can see here, the main idea is when it rains, there's not a lot of soil available for the water to go into, and so you end up with a lot of flooding. And then on the other side, you have a lot of drought, which is the more common state of urban soils, just because there's not as much soil volume to even hold moisture. Another thing, as I touched on briefly before about impervious surface, is if you have more impervious surface, you have to apply more winter salt to make surfaces safe for both pedestrians and for vehicular traffic. So here on the right, you see this is data from the New York open data that I pulled off from there. And as you can see, this New York City is a pretty big city, right? So we use a lot of salt to make our surfaces safe. And this is just from 2016 and onward, but you could see each year it's hundreds of thousands of tons of salt that we're applying to our surfaces. So despite all these things that are changed in cities, there's some wild city plants that are pretty tough. Some of these plant species have actually reached global distributions by being successful in human-dominated landscapes. And there is some evidence that some plant species are actually adapting to life in cities. So urban evolution studies have become more frequent over the last several decades. This is a nice schematic from Johnson and Munchi-Sal that kind of summarizes all of the efforts that have occurred throughout the world in studying urbanization. The colored dots refer to human population density, and then the blue organisms here are commensal species. But the main take-home message I want you to see from this slide is how many studies are actually focused on animals and how few studies have to date been focused on plants. So all of these studies together, we have now some very strong evidence that cities are driving the evolution of across taxa. So there's a book that's called Darwin Comes to Town that kind of summarizes some of these highlights. But there's other examples like white-footed mice having higher immune resistance and tolerance to heavy metals in cities as versus outside of cities, spiders getting bigger in cities and being attracted to artificial light sources, and lizards having stickier feet so that they can crawl on artificial surfaces better. And yes, even some evidence that plants are evolving in response to urbanization. So in the city, plants perform a lot of ecosystem services for humans. They improve air quality. They help mitigate noise. They support the biodiversity of other taxa. They prevent soil erosion. They provide cooling effects, and they have a pivotal role in nutrient cycling. So it's important to understand how plants will adapt in an increasingly urbanized world. They provide benefits to humans in urban settings. Urban areas are ridiculously complex ecosystems. Urban evolution, even though there has increased a lot in the recent decades, it's still a relatively new field, and most studies so far have focused on animals. And there is evidence that urbanization can drive plant evolution via natural selection. So we need more studies, particularly field and laboratory studies, to test for this local adaptation and to identify what environmental factors are driving this selection, because there's a lot of environmental factors. I just talked about a few of them on the previous slides. And so we need to start looking at each of these environmental factors to kind of understand the bigger picture. So that brings us to my research, which has 3 objectives. So I want to quantify the relative fitness of urban and rural populations of wild plants in New York City. Determine if those populations are locally adapted to conditions along the urban-rural gradient. And identify which environmental factors are driving selection. So if they are locally adapted along the gradient, what environmental factor is driving that? So as Dan mentioned, my cute charismatic species, this is Capsella bursipastoris, or shepherd's purse. Shepherd's purse is a relative of Arabidopsis thaliana, which is the plant study organism, which we know the most about. And it's a very, very, very small weed. It's less than 1 meter tall. And the majority of the plant body exists as this basal rosette of leaves at the base of the plant. And one plant individual can produce thousands of seeds. And they predominantly self-pollinate, which means that you only need one plant individual to make new plant individuals. And it frequently occurs in habitats that are very disturbed. So for example, sidewalk cracks, street tree pits, gardens, and even crop fields. In terms of the history of plant evolution, it's a relatively recent species. It's only estimated to be about 100 to 300,000 years old, and it originated in Eastern Europe and Western Asia. But it's now one of the most common weeds worldwide. So this is showing you occurrence data from GBIF, and these are all the occurrence records of shepherd's purse. The species tends to be highly variable in certain traits. So this might be a way that it can locally adapt. For example, Infer and colleagues found early and late flowering genotypes, which means like early and late flowering plant individuals that occurred at northern and southern populations of Europe, so across a latitudinal gradient. There's also a lot of ecotypic variation in leaf shape. So it demonstrates leaf polymorphism, and has been shown that these polymorphic traits can be conserved at the population level. So because of these things, it brings us back to our main question: is urbanization driving the adaptive evolution of shepherd's purse populations in New York City? And part of the reason I suspect that it is because of just where it occurs in cities. It's occurring in areas that are just prone to high amounts of abiotic stress. So the study's main hypothesis is that the urban populations of shepherd's purse have undergone adaptive evolution in abiotic stress tolerance because they've been exposed to it for so long over generations. And the abiotic stress in particular that I'm talking about is increased soil salinity and then episodic drought and flooding. So I would predict that plants from urban populations will demonstrate higher relative fitness, so be better fit, than rural populations when grown in both urban environments, but also under urban conditions. And the way I'm going to test this, or the way I did test this, is by using reciprocal transplant experiment and then laboratory abiotic stress trials. Okay, the 3 chapters of the thesis, we're going to go through them each today. But the main thing I want to draw attention to in going through this slide is that the first chapter is really focused on trying to see if there's evidence for the plant locally adapting in the real world, Whereas the second 2 chapters are in a controlled environment in the lab where I have more control and I'm manipulating a given environmental stress factor. So chapter 1, happening in the real world. Chapter 2 and 3, happening in a controlled setting in the lab. All right, let's dive into part 1. So part 1, is Capsella bursipisaurus locally adapted to urban environments? So I hypothesize they are. And I predict that the urban populations will have higher relative fitness when they're grown in urban environments compared to rural environments. And I'm going to use the reciprocal transplant experiment to test that. So just in case you are not familiar with the reciprocal transplant experiment, it's an experiment that transplants plants into reciprocal areas. So it's a field study, and what it allows you to do, it allows you to directly test for local adaptation in plant populations. So for example, here's a schematic, and you can see each of these. So I guess I can annotate. I'm very good at. So like here's an urban population, here's a rural population, right? During a reciprocal transplant, I'm going to take individuals that are from this urban population and I'm going to plant them right back at that same population. And then I'm going to plant some of those plant individuals at a rural population. And then likewise, plant individuals from this population are going to be planted at the rural population, the same population, and then some individuals at the urban population. Sorry, urban area. And if plants are locally adapted, they will have higher relative fitness at their origin home site. So what that means is if I take plants from this urban location and I plant them at that same location again, and they do better there than when I plant them here, there's evidence that they are locally adapted to that site. Okay, so how did I do this? So here's a map of our region with that impervious surface layer put on top. And so what I did is I basically established to look for any population within 60 kilometers from Times Square during a certain time period. And I quantified urbanization as percent impervious surface that I've already talked about before using QGIS. And so after doing surveys, I ended up collecting seeds from 360 plants in both urban and rural, or AKA less urban populations, and a total of 24 populations, which are shown here in the black dots. From those 24 populations, I selected 8 sites, 4 urban sites and 4 rural or less urban sites. And I picked those sites based on germination trials that I tested the viability of the seeds, safety and accessibility to the site, because I was doing all of this work majorly on my own, so I wanted it to be a safe place that I could actually access, and based on its impervious surface. So a site was designated as urban if it had more than 70% impervious surface, and rural or less urban if it had less than 50%. And before I talk about that a little bit more, I'm just showing you on the right here, so you can see that the colored dots, the dots with the same colors, are a pair. So you might look at 70% and 50% and say, like, well, that doesn't seem that different, Rebecca. Well, so that's just the bins that they are placed in, but if you look at the actual impervious surface values, the majority of the rural sites are actually much less than 50%. So here in this first example, site 1 and site 2, the rural site has 24%. Site 3 and site 4, the rural site has 33%. Site 5 and site 6 has less than 10%. And then the site 8 is the one where it's just a little closer to 50. So I took those seeds and I germinated seedlings in the vegetation room at CKB. And when the seedlings were 3 weeks old, I brought them to Brooklyn to leave them outside in germination hoods to help them harden off and transition from life in the lab to life in the real world. And I planted a total of 168 seedlings at the reciprocal transplant sites. So there are around 20 seedlings per site, 10 from that site and 10 from its paired site. And I had a lot of nice help with planting out the sites, not only from my advisor, Dan, but also from my friend Dennis and Lynn. And then this picture on the top left here is me planting the very first seedling. So I always include it. So you might wonder what these sites look like. Well, this is an example of one of those sites. So on the left, you have a less urban or rural site in Yonkers. And on the right, you have the urban site right in Williamsburg, Brooklyn. So when I planted the plants out, I took a bunch of site data, so different conditions like soil moisture and soil compaction, things like that on the planting days. And then I revisited every site 13 times during this period from March to June to record fitness data of the 20 plants at each site. So that included leaf length and number, plant height and width, flower number, seed pod number, and mortality, whether or not they were alive or dead. For statistical analysis, so we use mixed model analysis, and how that works is there's certain things that are fixed effects. So in our case, we have urban plant type or rural plant type, planting location, whether it's home or whether it's away, and the interaction of those things. And then we have some random effects, which are the parent plant that the seed came from and the paired site. And what we would do is we would fit that full model and then reduce the model by removing the random effects one at a time. And then compare those results to select the best fitting model using AIC and compared responses using F-test and orthogonal contrasts. So the idea here is if plants have higher fitness at their home sites compared to away sites, it's evidence for local adaptation. Just because I'm talking a lot. So let's look at some results. So the urban sites had higher average air temperatures, which we would expect given our urban heat island effect, which talked about higher air temperatures toward the beginning of the talk. They had pet excrement and garbage in 100% of the sites, and they had higher soil compaction compared to the rural sites. Rural sites had much higher greenery cover, and the pet excrement and garbage in 50% of the sites. Similar average values at both sites were things like organic litter, bare soil cover, canopy cover, soil temperature, soil moisture, and average metal concentration. So we took, I did soil samples to do heavy metal analysis of my sites. And on average, there was no clear difference between urban and rural sites. I will say, however, that each site had almost a very unique metal profile. And so there was a lot of variance and very wide concentration ranges. All right, more results. So both plants live significantly shorter lives at urban sites. So we're going to see a few graphs that look like this, so I'll kind of walk you through it. It has site type on the bottom, so home versus away, and then plant type on the top, rural on the left, urban on the right, and in this case, survival is on the Y. And so you could see rural plants when they were away at urban sites, they lived much shorter lives. And urban plants, when they were home at urban sites, they lived much shorter lives. Part of this was because of random events at the urban sites. So for example, on the top right here in B, you see a site, that Williamsburg tree pit site. So 3 days after I planted the seedlings out there, that site got run over by a truck and effectively killed almost all of my plants at that site. So I did a few days later get to plant some more seedlings back there, but that was unexpected, couldn't plan for a truck. And then the other thing, unfortunately too, is one of my sites also became an encampment, such that all of my plants had a structure built upon them. And even though I lost the data from the site, obviously, like this is a bigger It made me humble, I will say. Okay, urban plants began flowering much earlier at urban sites. So here again, similar type of graph. Rural plants, they kind of perform the same in terms of flowering time, whether they were home or away. But urban plants took a significantly longer time to begin flowering when they were at rural sites, and significantly sooner when they were at urban sites. Both plant types had longer reproductive durations at home sites, demonstrating that they are indeed locally adapted. So here you go, you have reproductive duration, and that means from the day of first flowering through the end of the reproductive period. And as you can see, both rural and urban plants, when they were home, had much, much longer reproductive durations. And urban plants had much higher fecundity. They produced way more seed pods at home sites, demonstrating that the urban plant adaptation translates directly to fitness in this case. So you can see the plot on the left. The urban plants, when they were in urban sites, produced 4 times more seed pods than when they were away. And the rural plants produced roughly the same amount. On the right, you have a detail of the seed pod on the bottom, and then the detail of the infructescence in B. So we have support for our hypothesis of local adaptation. Urban plants flowered sooner at urban sites. Urban plants produced significantly more seed pods at urban sites, and both plant types had significantly longer reproductive duration at their home sites. So that is very strong support and shows that these plants are locally adapted. But now it comes the next task, which is to figure out which environmental factor is primarily responsible. So is it air or ground temperature? Is it differences in water regimes, right? During the trials, we saw that urban soils indeed have higher soil compaction. And if your soil is more compacted, it's going to reduce how much water can actually get into it, which is going to increase flooding and drought. And we did observe both flooding and drought at our urban sites during the reciprocal transplant experiment. And it could also just be due to soil pollution. I mean, the urban sites did have slightly higher sodium than the rural sites. But again, the variation in the metal concentration at the site level was so wide. And so there's a potential possibility of local adaptation to that home site metal profile. Okay. So, evidence for local adaptation in the New York City metropolitan area of Capsella versipastores. Yes. So now we have to figure out, okay, which environmental factor is responsible, right? So these are the lab trials. And so the first one we wanted to test is, are urban plants better adapted to hydrological variability found in urban areas, right? So water stress. As I mentioned, cities have a high proportion impervious surface. And so this leads to more frequent drought and more periodic flooding, right? And I think I've already kind of explained how that works, but less soil volume, less ability to hold moisture leads for drought, and then compacted soil and also compacted soils. And then for flooding, compacted soils, and then lack of soils as well, tons of flooding whenever it rains. And so just to remind you about what the habit or form of my species is it's small. It's a small plant, and the majority of the plant exists at the basal area, right at the base. And so it's vulnerable to even mild flooding events. And it lives in soils that are prone to drought. You have higher radiant heat from the paved surfaces around these soils that's causing higher air temperatures. You have compaction, and you have less volume, so prone to drought. And as we already talked about, there's a ton of impervious surfaces in New York City, so we would expect more drought and floods. So, and as I a little bit mentioned before about shepherd's purse has these like variable leaf traits, right? So these are potential things that can be selected for to promote drought tolerance in particular. So they have different surface areas, different degrees of lobing. If you have less surface area, you will have less water vapor loss. And if you had a wide surface area, different degrees of thickness, thicker leaves might be able to hold water better than thinner leaves, different things. And so Infer and colleagues did a study where they subjected different members of Capsella to water stress. And for Bursa pastoris, they did see that when it was subjected to water stress, you did have denser mesophyll cells and less extracellular space. So I'm just going to see if I can annotate real quick. So down here is without water stress. And you can see like there's these big openings. These are big openings here. Those are just space. Those are not cells. Whereas up here, there's a lot less openings, right? The cells are denser together. The other interesting thing they saw is underwater stress, Capsella bursa pastoris actually created two, here, I guess I'll annotate again, 2 layers of palisade cells. Not that great at annotating, as versus without water stress. And those play a role in light absorption. Oh, there we go. So what that would mean is if you had this higher water stress and this was what your leaves were doing, you would expect those leaves to have higher leaf dry matter content and lower specific leaf area. And so what are those? Well, let's talk about them. So leaf dry matter content is the ratio of leaf dry mass to fresh mass. So you take a leaf off of a plant, you weigh it, and then you dry it for like 72 hours for it to lose all of the water. And what leaf dry matter content tells you is an indicator of how the plant is using resources. So if you have low leaf dry matter content, you're growing the leaves faster, they're less robust. Whereas if you have a high leaf dry matter content, you're putting more investment into your tissues, so you have better conservation of resources. Specific leaf area is the area of a leaf per its unit mass. So it's positively correlated with relative growth rate. So if you had higher values of SLA, it would indicate, it would correlate with more growth. And you can also use it as an indicator for resource use, right? So if you have low SLA, you have less leaf area per your carbon investment and you're less efficient. Whereas if you have high SLA, you have more leaf area per your carbon investment and you're more efficient. Okay. So these are the lab trials, remember. So our hypothesis is that urban populations are locally adapted to these urban water stress regimes, right? And so we would predict that compared to the rural plants, the urban plants are going to have higher relative fitness, right? Longer reproductive duration, more seed pods, bigger plant sizes when they're grown in all the water stress treatments. They're going to have a higher tolerance to water stress, so they're going to live longer when subjected to water stress, and have leaf phenotypes or leaf types that are better able to mitigate the effects of water stress. So as I mentioned on the previous slide, we would expect, compared to the rural plants, that the urban plants would have higher leaf dry matter content, lower specific leaf area, fewer stomata, which we haven't talked about yet, but we will, and more trichomes. And we're going to test these things using the laboratory stress trials. So here we are in the vegetation room. We ran a total of 3 trials because of the space that we had and things like that. So, and we wanted to increase our sample size number, so we ran 3. We used a total of 392 plants from 10 urban populations and 9 rural populations that represented 80 parent plants. And these trials involve different degrees of soil drying. So we quantified watering by weight. There's different ways to quantify it, but this was the way that we thought would be best for our study. And namely, the plant is very small. And so putting a soil probe, a soil moisture probe into the soil over and over and over again, I just didn't feel comfortable. I thought it could cause too much abiotic mechanical damage. So we went with weight. Each plant was planted in the same pot type with the same amount of soil and then weighed to 300 grams to start the trial. So there are 4 treatment types that we used in the experiment, there's a control where plants are watered gently 3 times a week to 300 grams. So they're never super, super moist, and they're never dry. They're kind of just nice all the time, optimal conditions. Drought plants, the water is withheld for 1 week, and then we do a partial rewater to 200 grams, and that occurs every week. The reason we did that is because we wanted to subject them to drought, but we also wanted them to reach reproductive stage so we can compare the seed pods and flowers. And so this is a method people use with this partial rewater to extend the period of keeping the plant alive and subjecting it to drought. Flood, the plants were submerged by more than half for a week, allowed to partial dry for 1 day and repeat. Same logic there about the partial redry to extend their period of life. And then a treatment that is like a cyclic drought flood. So the water is withheld for a week, plants are rewatered to 300 grams, then submerged for 50% for another week, and then repeat. So in trials 1 and 2, included all 4 of these treatments. Trial 3, we only did drought and control because drought in the first 2 trials seemed to be the stress that was causing the biggest change. Okay. During the trials, we recorded these different fitness measurements, so leaf length and number, what the state of the leaves were, the basal area, which again is that lower, so I have the mouse pointing on it now. This is that basal area here, the lower part of the plant. Flower number, seed pod number, and mortality. And so for the first trial, we took data a lot at the very beginning because we didn't know how fast we were going to see a response. So the first trial ran 6 weeks, and we recorded this fitness data on the day we repotted the plants, at day 0 or the start date of the trial, and then on these other days, 3, 5, 10, etc. during that first trial. During the second and third trial, the second trial ran 15 weeks, third trial ran 7 weeks. We didn't take it as frequently, and so because we realized we can do it weekly, and we did it weekly during those experiments. Okay. So we're talking about water stress, right? So there's certain leaf traits that help plants maintain biological activity during water stress. And they do this by helping the plant keep water that it has by sequestering it in parts of its plant body or by preventing water loss through other mechanisms. These are the types of leaf traits that can help a plant do better in water stress. Denser leaves, waxier cuticles, increased trichomes, decreased stomata, and decreased leaf area. And so what we did is we harvested for the third trial, that control versus drought, we harvested leaves from living plants that had at least 3 healthy leaves at the end of the third trial. It ended up being 50 plants. So we already talked about leaf dry matter content, but this is one of the traits that we measured. And the idea is that plants with the higher leaf dry matter content can withstand the water stress because they can withstand the lower negative water potential in the plant, in the leaf, and therefore maintain physiological activity during drought. So we would predict that urban plants would have higher leaf dry matter content. Specific leaf area, which we've already covered, remember it scales positively and linearly with growth rate. So plants with lower SLA have less surface area per carbon investment. So they have smaller leaves, which could lead to less water loss. And so we would predict that urban plants would have lower SLA in drought. And then stomatal density. So stomata, and if Dr. Casper is watching, I'm sorry, I'm not calling it stomatal apparatus, but it's just for time. But so for stomatal density, these are holes on the opening of the leaf. And what they do, as you can see in this little schematic on the bottom left, when they're open, they allow CO2 to come in. CO2 is needed in photosynthesis, how plants make their food. So opening the stomata lets CO2 in, but when it's open, this also releases water vapor and oxygen, which are products of photosynthesis. So if you're open more, or you have more of them, you would have more water loss. So plants with fewer stomata have a less potential for water loss. And so we predict that urban plants will have fewer stomata in drought. Okay. Trichome density. So trichomes are hairs that are on the, in this case, the leaf surface. They can be on other plant body parts too, but in this case, we're talking trichomes on leaves. The trichomes have all different types of purposes, but one of the functions that trichomes do is they help keep leaf temperature cool and help reflect sunlight. And so that can help the plant keep more water. So plants with more trichomes have less potential for water loss and therefore predict that urban plants will have more trichomes in drought. So this is an image that I took of one of our plants. So this is a microscope slide which had trichomes on it. So the species produces two types of trichomes on one individual, just like Arabidopsis thaliana. Stellate trichomes are star-shaped trichomes, and then spike-like trichomes, which I call single-haired trichomes. So these are both from the same leaf. They're on the same microscope slide. I just removed the background, but this gives you an idea of what their sizes are. So the spike ones grow vertically from the leaf surface. The stellate or star-shaped ones grow horizontally. They branch out parallel to the leaf surface. This I'm just showing you to show you there's a lot of variation in plants. Some plants have, so you have one individual on the top. The images on the right side have been inverted and the contrast has been increased so you can see the trichomes better. But the point of this slide is that some plants have way more star-shaped trichomes than other plants, and some plants have more single-haired trichomes than other plants. And so that's why we wanted to count not only the density of trichomes, but also the density of the two types. Statistical analysis is similar to the type that we used before, right? We fitted the full model with the plant type, treatment type, and the interaction of those things. And then in this case, the random effects were seed collection population, treatment block, and parent plant. We reduced the full model by removing random effects one at a time, tested those for the best fitting model using AIC, and then compared responses using F-test and orthogonal contrast. And the only other thing we did in this case is we used a principal component analysis on leaf traits and plant size. Okay, now we'll get into results. So there was no mortality. No plants died in any of the trials. And plants from population, oh, right. So this is population 23. It actually started to flower before trials even began. So because of that, we don't include any plants from that population. Even if they didn't flower, we just didn't include that population at all in this data. But once that population is taken out, very, very few plants flowered, only about 13% across all the trials. And of the few plants that did flower, there were no clear trends. Okay, so now we'll look at basal area, right? So final plant size. This is our final plant size of plants that survived the water stress treatments, okay? So this is from the first trial. And as you can see, so you have urban on the right, you have rural on the left, and you have the treatments on the x-axis, and then final plant size on the y. And so for urban plants, you could see they were basically unaffected, right? They grew the same whether they were in the control or in the drought, flood, and flood. I should mention that these error bars are 95% confidence intervals. Whereas with the rural plants, they did seem to have their growth impacted by drought, right? So the drought was significantly lower than the controls. So this is for the first trial. For the second trial, we basically saw a treatment effect, meaning they grew based on how the treatments were, but there's no, so the main part here is urban plants unaffected. This is the first one I showed you. Urban plants unaffected, rural plants affected only in drought, right? Plant size decreased in drought. Here, urban plant and rural plants pretty much respond the same. There's some trend here that maybe the rural plants are slightly larger in the drought treatments, which is different than the first trial, right? So it's a substantial treatment effect except for that drought one in rural. And for the final plant size of the third trial, we didn't see anything. No significant effects. Everything grew the same. So to summarize the final plant sizes, there was a lot of variability, and so we can't really gather any clear patterns across all three trials, right? No clear conclusions there. So much variability, plants overall unaffected. All right. So now let's talk about the leaf traits, right? So we had predicted that urban plants would have higher leaf dry matter content than rural plants. And as you can see on the left here, both urban and rural plants responded the same. They both responded in having higher leaf dry matter content under drought. And then for SLA, we predicted or expected that urban plants would have lower SLA compared to rural plants, but we did not see that. SLA was the equivalent for both plant types in both treatments. This is stomatal density. So we had predicted that urban plants would have fewer stomatal density under drought compared to rural. And indeed, we did see that here. So rural plants actually had an increase in stomata under drought conditions, which is interesting. Whereas urban plants had a fewer stomata and drought, which is what we expected to see. This is the comparison of the trichomes. On the left, you have single-haired trichomes. On the right, you have star-haired trichomes. So we had predicted that urban plants would have less trichomes of either type under drought conditions. But again, both rural and urban plants had the same response to the drought treatment. So in the single hairs, we did see a decrease in single-haired trichomes under drought for both plants. But in star hairs, we actually saw an increase in star hairs under drought conditions. These are the principal component analysis we ran to compare traits and final plant size of surviving plants. So on the left, you have a PCA of all the leaf traits for the control plants. And on the right, you have a PCA of the leaf traits for the drought plants. In both cases, that first axis, which is driven by leaf dry matter content and specific leaf area, explains the majority of the trait variation that we have in both cases, whereas the second axis represents far less of the trait variation. So we took these and we put them, tested them models to see if this was something that traits could predict the final size of our plants. And for drought, not control, but for drought, we did find that our model suggests that the final plant size can be predicted by the PC1 values. So those leaf dry matter content and specific leaf area can predict final size of plants in drought. I should mention that in our best fitting model, it included treatment block as a random effect. So you can see here on the right is block. And for all of these treatment blocks, except for 16, right, you see a positive relationship with these leaf traits and growth. Whoops. Okay. So we'll discuss the water stress trial. So we expected the urban plants to have higher relative fitness and survival under water stress, but we did not see that, right? Urban and rural plants responded similarly, and there was no mortality and very low flower production and seed pod production, so we couldn't statistically analyze those. But overall, both plants appeared basically unaffected by water stress in terms of plant size. But again, our first trial, we saw urban plants unaffected by water stress and rural plants unaffected by the flood and drought flood treatments. but strongly impacted by drought. But the second and third trials were inconclusive, and so therefore we didn't see any clear patterns, except potentially very unaffected by water stress. We also expected urban plants to have leaf phenotypes that better mitigate water stress. But again, both plant types responded similarly. So this shows that the species is very plastic in leaf trait response to drought. You know, urban and rural plants both had a definite change when drought occurred, except for SLA. But because they're so plastic in this response to drought, this could explain why the plants are basically unaffected by water stress, because they make changes in order to compensate for that water stress. So plants that were grown in drought were larger if they had higher leaf dry matter content. That was for both plant types. Lower specific leaf area. This is based on the PCA analysis. And fewer stomata. That was for the urban plants. Okay. Some factors that may have impacted the results that we got. So we had low flowering and low seed pod production, and this could be because the trials had to end due to time constraints. So if we were to allow the trials to continue for a much longer time, we may have seen higher flowering and seed pod production. The other thing is there was no mortality. All the plants survived. So this could indicate that the water stress treatments were just not stressful enough. So in the first part, we did this reciprocal transplant experiment, and we did find evidence for local adaptation in these plants, right? Then we bring the plants into the lab, into a controlled setting where we can control for everything and change the water stress that they're dealing with. And we didn't see any clear trend there. So now the question is, well, it's probably, based on our results, probably not water stress driving this local adaptation. So what about salt stress? And so that's what this next part is about, the impact of salt stress on growth and survival of Capsella versipistors. As a reminder, right, cities have a high proportion of impervious surface, which means in areas that it's cold, you have higher winter salt application. So this is New York City. Again, you saw this from the beginning in the hundreds of thousands of tons of salt we use each year. And we use a lot of it, a lot of impervious surface. Just to give you some background, so this is de-icing salt application since around 1950. I guess it's 1940. And you have total salt consumption and then salt used for roadway de-icing in the darker blue color. And as you can see, as cities have increased, roads have increased, and we have seen a real dramatic rise. So this is metric tons in millions of salt used to de-ice roadways. And the majority of the salt that's used, 98% of the salt that's used, is sodium chloride. So we don't know, there's not a lot known about salt and Capsella versipistoris, but we do know that the species is capable of living in substrates with high toxic iron concentrations. So there's a potential there if it can survive living in high levels of toxic ion concentrations. Well, salt is basically a toxic ion in the soil, so at certain concentrations. So there's a potential there for selection. In this paper, Axway and colleagues showed that Capsella versipistoris can maybe be used as a biomonitor of heavy metals. And I should point out that heavy metal ions in the soil cause similar ion stress that salt would cause when it's in the soil. And what they found is that there are evidence for the species to have higher levels of ion concentrations when ion level increased in the substrate. So I pulled this one example from their paper, is showing lead in the leaves of Capsella versipistoris as lead in the soil increases. So as lead in the soil increases, lead in the leaves is also increasing. So the lead is being absorbed at the roots and making it all the way up to the leaves. So if it could absorb lead, maybe it could absorb salt. And so let's see. So we have the hypothesis that the species is locally adapted to, the urban populations are locally adapted to salt stress. And compared to the rural plants, We think that urban plants will have higher relative fitness in all the salt treatments, demonstrate higher tolerance to salt stress, right, live longer, and have leaf phenotypes that better mitigate the effects of salt stress. And these are similar, actually the same types of predictions we would make for the water stress because of the way that salt responds to the plant. So higher leaf drier matter content, lower SLA, fewer stomata, and more trichomes. And we're going to test this using laboratory salt stress trial. All right. So there, in the case of the salt stress trial, we had 4 water treatments for treatment blocks 1 through 6. And these water treatments were 0, 50, 100, and 150 millimolar NaCl. However, this resulted in really high mortality rates, really high. So we therefore lowered the concentrations for the subsequent treatment blocks 7 through 9, such that they had 0, 20, 40, and 60 millimolar NaCl. So we used a total of 288 seedlings for these salt stress trials from 11 urban populations, 10 rural populations, representing 36 parent plants. And each treatment block included 4 trays, 1 tray per treatment, 32 plants, or 8 per treatment, 4 urban and 4 rural populations with 2 parent plants representing each population. Okay, recorded similar data that we did in the water stress trials, leaf number and length, flower number, seed pod number, mortality. And then at the very end of the trials, we measured the actual salinity content of the plants that lived and plants that died. And we took fitness data on repot day and the first day of the trial, which is day 0, and then every week. For 5 weeks, we took data. The way we applied the treatment was we would pre-mix 4 liters of the treatment solution to get the right millimolar concentration. And we would pour that into the correct trays so that the treatment water was placed at the bottom of the trays so that to prevent water from spilling onto the leaf or the plant body, right? So we bottom watered them. The salt doesn't go away, the salt stays in the system, and so we would just re-water with fresh water. So every week we would add 4 liters of fresh water to the system, and the same level of salt would just be maintained because it doesn't evaporate. And to measure the leaf traits, we did the same thing when that we took 86 plants that had at least 3 healthy leaves at the end of the 3rd trial and measured these leaf traits, leaf dry matter content, specific leaf area, stomatal density, and trichome density. And as I mentioned, salt stress can cause this kind of physiological drought in that it makes it so that even if there is water present, the plant can't absorb the water because the salt ions are holding onto it. So therefore, we predicted that these are what we would expect the urban plants to have: higher leaf dry matter content, lower SLA, fewer stomata, and more trichomes. We use linear and generalized linear model analysis. So similar to what we've already gone through, we fitted that full model with the different fixed and random effects, reduced the full model by removing each random effect, selected the best fitting model using AIC, and then compared the responses with F-tests, likelihood ratio tests, and orthogonal contrast. So we had a lot of mortality this time. So many plants died. About 38% of plants died during the trial, most of which were in the treatments 50, 100, and 150 millimolar NaCl. So in this table, you can see of the plants that died at 50 millimolar, more of them were urban plants, right? So you had 57.9% of the plants that died in 50 millimolar were urban. For 100, nearly twice as many plants that died were urban at 100 millimolar. And at 150 millimolar, very similar, about half of the plants that died were from either plant type. And we had very little to no mortality in 0, 20, 40, and 60 millimolar NaCl. So because we had such little mortality on one end and then so much mortality on the other end, we needed to group our treatments together to improve model convergence. So we had a low salt category, a medium salt category, and a high salt category. And here are the results. So both plant types have decreased probability of survival as salt stress increases, right? Salt kills. Salt kills. And the rural plants actually survived slightly better. But in general, the take-home message from this analysis is salt kills. And again, very, very few plants produce flowers and even fewer produce seed pods. And again, population 23 began flowering before the trials began. So we don't include that problem. And that's a whole nother study that needs to happen with that, with this flowering time thing, because this is obviously conserved. But once we remove that population, very few flowers, only 3% out of the trial flowered. So again, couldn't really analyze that. So again, we're looking at final plant size for the survivors. So this is final plant size of all of the, you have rural on the left, urban on the right. And as you can see, both plant types responded similarly, right? The survivors of both plant types had the same reaction or response to the different salinity concentrations. What was I going to say? Oh, right. But the weird, I mean, the thing here, though, to take away is that this is final plant size. And just look at your high values, right? Your 100 and your 150 millimolar NaCl. Plants that survived those treatments were significantly larger than plants in the controls. So even though salt kills really effectively at those concentrations, the plants that survived were significantly larger than all the other plants. So leaf dry matter content, we thought that we would see a increase in leaf dry matter content, but in fact, in urban plants, but in fact, we see a decrease leaf dry matter content for both plant types. We thought we would see a decrease in SLA in urban plants, but again, here we see an increase in SLA for both plant types. We thought urban plants would have less stomata compared to rural plants as salinity increased, and indeed, both plant types have less stomata as salinity increases. We'll talk about that. We'll compare them in a second. This is an interesting result. So here we expected urban plants to have more trichomes as salt increases. And instead, and urban plants do have more trichomes and single hair trichomes as salinity increases, whereas rural plants have a decrease here. And both plant types have fewer stellate trichomes as salinity increases, both plant types, with rural slightly less. So let's talk about this a little bit. So we expected the urban plants to have higher relative fitness and survival, but mortality was the same, basically, except for at that 100 millimolar. But otherwise, mortality, both plant types had high mortality at high concentrations and low mortality at low concentrations. Again, low flower and seed pod production for both plant types. The model did suggest that rural plants might have a slight better survival rate as salt increases. And to me, the most interesting result is the final plant size. So plants that survived high concentrations of salt were significantly larger plants, 2 times larger than control plants for both plant types. And so even though salt kills, for these plants, this is an indication that these plants are okay with salt, right? They're adapted to salt because they had, not only did they survive, but they grew twice as big. With the leaf phenotypes, we expected urban plants to have the ones we have outlined before, right? That are better mitigate salt stress. But both plant types responded similarly. again, showing that the species is very plastic in how it responds in leaf trait to salt stress, very plastic response. And it could explain, being so plastic could be an advantage, and that could explain why those plants survived. So maybe the plants that survived the salt stress are the most plastic of all, and so they were able to overcome that stress. But this is what I really wanted to get to, because this is so interesting. So the plants respond differently to salt stress and drought, which indicates something else is happening here, because a lot of the literature suggests that salt stress and drought stress have a similar response in plants. But here we saw a lot of differences. So for example, in the salt stress, leaf dry matter content as salinity increased, decreased, right? So they had lower leaf dry matter content as salinity went up. So to me, this kind of seems like the leaves might be sequestering water because they have more water per volume. Whereas in drought, leaf dry matter content, okay, hold on. So sorry, leaf dry matter, that's a typo. Leaf dry matter content increased in drought, that's a typo, sorry. Decreased in salt, increased in drought. For SLA, in this salt stress trial, it increased. So the leaves got thinner. Whereas in the drought trials, the SLA was not affected by drought treatment. In salt, stomatal density decreased for both plant types, but in the drought conditions, rural plants had an increase in stomata. In salt, the stellate trichome density decreased for both plant types, but in drought, both plant types had increased stellate trichome density, so the opposite. Single hair trichome density decreased for urban plants with salt, and increased for rural plants in salt, whereas in drought, both plant types had a decrease in single hair trichome. So but in salt, it increases in rural plants. So that's something we gotta look into more. And so the other thing I want to point out here is plants are taking up salt. They're obviously not preventing ion entry at the roots, because if they were preventing ion entry at the roots, they would respond way more like as if they were in a drought condition. But that's not what's happening. They are taking up the salt. And we actually saw, remember, our plants are bottom watered, but on some surviving plants, we actually saw salt being excreted from some of the leaves. So they're definitely taking up the salt. We need to talk about the factors that might have impacted our results. So we did again have low flowering seed pod production. And again, maybe if the trials had continued longer, we may have had more flowering in seed pods. Also, something is off with our results for 60 millimolar, because based on the 50, 100, and 150 mortality rate, 60 millimolar had a very low mortality rate. So something is off there. It should have been higher, or maybe something was off with 50 millimolar, because either way, there's something going on there, and something is off because there's a dip in mortality there. And the species is surprisingly salt sensitive, even though it occurs in street tree pits and sidewalk cracks and roadsides. But it's surprisingly salt sensitive. However, if they're able to survive the salt, they have larger plant sizes, and so survivors seem to be adapted to salt. Okay, almost there. So for the reciprocal transplant experiments, right, we had support. We had support for hypothesis of local adaptation. The urban plants had significantly higher relative fitness at urban sites. Both plants had significantly longer reproductive duration at their home sites. So we have, in that real world experiment where we have no control over anything except showing up to take data, we have the support for that local adaptation. But we bring it into the lab to test these environmental factors where we have all of our control, and we don't see the clear patterns that we were expecting to see, right? So For water stress, our hypothesis that urban adaptation is via water stress in urban plants is not supported. Trials were overall inconclusive. Although the urban plants did produce fewer stomata under drought conditions. And we did find that you could use leaf traits, particularly leaf dry matter content and surface specific leaf area to predict final size of drought plants. And in the lab where we have all the control and we're manipulating the salt environmental factor, we also don't have support for the hypothesis that the adaptation we saw during the reciprocal transplant experiments is due to salt. We had similar mortality for both plants. Rural plants even might have a better survival under salt stress based on our model. Species is very salt sensitive, which surprised me. But the plants that do survive have a significantly larger size than control plants. And another surprise was the difference in leaf trait responses. It's just the plants are so plastic and they react so much to this stress, and it's a very clear reaction, but depending on what the stress is, it's a very different reaction in leaf traits. Before I go forward here and close out, I have to thank, first of all, I have to thank my advisor, Dan, for being there for me and helping me through this and just putting up with me to some extent. But no, I really thank Dan. We've become great friends. And I want to thank the Bunker Lab, Sean and Caroline, my committee members, of course, Jessica Ware, Klaus Holzefeld, Philip Bard, and Emily Josephs, the collaborators we've had at NJIT and Fordham University. And of course, my family and my husband and my friends and everyone. But I really have to also thank my undergraduate researchers, Monica Solomon, Gabby Amparo, Stevens, Nemi, and Nancy. These undergrad researchers they've put so much work toward their independent projects and towards helping me run things in the lab. Monica is now off at dental school at Harvard. Stevens and Gabby will be graduating this semester, as I think Nancy is also graduating this semester. So they've just been so wonderful. And they helped with both the water stress trial and the salt stress trial. So their positive energy and enthusiasm in the lab really made life better, because it could be a little isolating in that vegetation room, I'll tell you what. And with that, I'm going to end it, and I thank you so much for being here. I really appreciate it.
Everyone should unmute and applaud.
God. Good job, Rebecca.
Well, we're done.
Woohoo!
Hi,
How do I see people?
Hi, Rebecca.
okay, so we will have another round of applause a little bit later, because that's just fun. It sounds good, actually. Better than enough. We're taking questions now, and as Dan said before, the question should come from the audience, not from the committee, because we will have a lot of time to ask questions afterwards. What I think we should do, you see that little, before you start talking, the little signal. Okay, please raise your hand, and I'm trying to call on you whenever. I hope I have an order here. So, raise your hand.
Hey, Klaus.
Yeah.
Dan here. We suggested that they type question into the chat box, and if you open up the if you close the participants and look at the chat, you should be able to scroll.
Then I get the timing as well. Yeah, you're right. Okay, so
Get them in order. Try that.
So, type the question, and I will call on you.
And also, if you're not asking a question right now, please remute your microphone.
Just press it.
Where did we lose Klaus?
Yes, you lost me, but I'm back.
Okay. Okay. Well, Amy Burkhoff typed a question in the chat box.
Okay, so answer it then.
Yeah. All right. Hold on. Let me get it here. So she said, why do you think the ratio of stellate hairs increased under drought stress? So I think that the stellate hairs, which are very, I'm gonna go back to this. I think it has to do with their ability to prevent water loss. So like I said in this slide, the stellate hairs are quite big, but they run horizontally. Basically, they're almost like little umbrellas, and so they're parallel to the leaf surface. So I think their ability to prevent water loss via wind, like excessive water vapor loss, is probably better than the spike-like trichomes, the single-haired trichomes. And I think the single-haired trichomes may actually play more of a role in insect, like in preventing herbivores. Here, let me just show you this picture here. Yeah, so like this single-haired trichomes, I think their role is less involved in water vapor loss, and I think the stellate trichomes do help prevent water loss. And then Haley had a question, but I don't know where.
Yeah, I figured it would be easier to just ask.
There we go. Yeah, sounds good.
So I kind of two questions. So first one's more of a general one. Does the soil salinity in the both urban and rural areas, does it decrease across the year, like between like say spring and fall? Like if you were to test soil in the spring, the summer, the fall, would you find decreasing salinity?
So that's a great question. So the key time of year that salt is used to apply is like your winter and spring times. And so I think you would have in general higher levels of soil salinity during the spring months. But that's also the time that shepherd's purse is the most active. So I might have forgotten to mention it's a cold season annual, which means that it's most active in the mid to late spring when we're still a lot of times using de-icing salt, but it's right during the end of winter too. So yeah, that's a good question. You definitely can have so via flooding events and things like that over the summer months, you probably could have a decrease in soil salinity over those summer months. But at that point, shepherd's purse is done.
Okay.
Great question.
And my second question was, when you're selecting parent plants to use for your lab experiments trials,
Mm-hmm.
I think there's a chance that some of the plant or did you select plants that were specific, like in specific plots or was it just random, or was it gosh, I don't know how to phrase this. I guess what I'm asking is, do you think that part of the reason that why you might have had that like funky lab data that kind of contradicted some of the things you saw in real life, was it, do you think, partially because of some of the parent plants you chose, that they might not have a good salt response based on their plot locations?
Sure. So that's another great question. So the way that I chose what parent plants to use for the lab trials was based on germination trials to just see, so whichever parent plants had previously germinated before in the germination trials. I also did include parent plants in the lab trials that were involved in the reciprocal transplant trials as well. So in some cases, in almost all the cases, the parent plants that made a lot of flowers and seed pods in the reciprocal transplant experiments never did flower or produce seed pods during the lab trials. So I know that those populations are capable, or sorry, I know that those parent plants are capable of producing a lot of flowers and a lot of seed pods, but they didn't in the lab trials. And I don't know if that's because the lab trials were too short, or if there was some other environmental condition in the lab that was preventing them from reaching that reproductive phase. There's also the potential that life in the city, like life out in the city, it's not just water stress and it's not just soil salinity, right? There's so much going on. There's pet excrement. There's so many environmental factors happening all at once. And so there's a chance that my lab individuals, compared to their life out on the streets, as it were, my lab individuals might have been in that cushy life because they're only having one environmental stress and they're used to having 100 environmental stresses. And so they may have said, oh, well, I'm released from stress. I can spend more time growing vegetative tissue. I don't need to go to flower yet. I'm living the good life. But that's also a possibility. But great questions, Haley. Thank you.
Okay, so the next question, I think, would be Jason, right?
Yeah.
Rebecca, you see that?
Yeah, let me pull it up here. Okay, general question. Do you think local adaptation to urbanization is likely to be a widespread phenomenon among plants or limited to a few species that thrive in disturbed locations? Is it affected at all by gene flow and from surrounding less urban areas? Great question. Okay, so if plant populations are existing in urban areas, so okay, I have to kind of pull this question apart a little bit. Plants have different life history strategies. Some plants require a biotic pollinator, right? Some plants require a bee or something like that. Some plants need to cross-pollinate and they have to have 2 plant individuals in order to have fertilization occur, right? Some plant species are able to pollinate themselves, like shepherd's purse, right? So that one plant individual can reproduce on its own. Plants are also vectored differently, right? Some plants are, for example, bird dispersed, right? Birds eat the seeds, fly around, and then deposit the seeds in different places. Some plants aren't, and they just drop seeds down, or the seeds can fly away on the wind a certain distance depending on the seeds, right? So it's such a big, it's like plants are not all created equal, right? They all have different life history strategies. And I actually think in order for us to understand evolution of plants in cities, we have to test plants that have all of these different life history strategies. We need to include plants that are stress tolerant and plants that are not stress tolerant. We need to include woody species and herbaceous species, and we need to include species that are animal dispersed, not animal dispersed, and self-compatible and not self-compatible, because they're all going to be affected by different things. So I guess with that all in mind, Jason, with that all in mind, I'll say, I do think that local adaptation to urbanization is likely a widespread phenomenon among plants. I do. I don't think it's limited to a few species that thrive in disturbed locations. I do think that plants that can survive in disturbed locations might have a pre-adapted advantage when they are in city conditions. But if plants are in the city and they're existing there over generations, they're definitely being impacted by urbanization over time. And I think local adaptation would occur in a broad amount of plant types. The thing, is it affected at all by gene flow from surrounding less urban areas? That's really going to depend on your pollination syndrome and your seed dispersal mechanism. So that's the easiest way I can answer that. I mean, think of shepherd's purse. Shepherd's purse is primarily selves. So you already have a great limitation on gene flow, that alone, right? So it's such a big loaded question. Oh, I have a question here. from whatever happened to the collaboration with the Fordham grad student with pigeons. Yeah, so she's here. Her name's Elizabeth. She's here. I have seen, actually, I'll show you. Great. I'm glad that Judy asked that. I can include this slide here. 1 second, it's at the end. Yeah, so, all right, it'll come in a second. Here we go. Yeah, here, I'll share it big. Yeah, so Judith Weiss, she's asking about pigeons, because here's a slide. This is from May. I don't know why the date's not on this slide. It's supposed to be on this slide, but basically, when I was doing the seed collections, I witnessed firsthand pigeons walking from shepherd's purse plant to shepherd's purse plant, consuming the seed pods completely off the plant. So on the left here, you see a shepherd's purse plant that's covered in seed pods. A pigeon was there for like 2 minutes and ate every single seed pod off of the plant. This is it walking away right there. That's the one. So that made me wonder if shepherd's purse could be bird dispersed, right? As particularly by common urban birds like pigeons. And so I do want to test this someday. I need to figure out how to test this the best way. But so Elizabeth Carlin works with, she's at Fordham University, and she works on studying pigeon evolution in cities. And so she was nice enough to let me come, and I actually did some pigeon dissections where I went through their innards to see if I could find any shepherd's purse seed pods. I did not find any that day, but those birds also had been baited by the airport, and so they were pretty much full of birdseed. Let's see. Here we have, how can you infer local adaptation by looking at plastic phenotypic traits? Why not use genetics? Great question, Angelo. The plan would be to ultimately do that. I would love to run population genetics on my species. Also, I would love to do RNA analysis to look at different stress responses. It is, as I mentioned, a very close relative to Arabidopsis thaliana, and the other members of Capsella genus have been sequenced. The genomes have been sequenced. So I would love to use genetics. I just couldn't afford to do that. Amy, you raised your hand?
I don't see a raised hand anymore. So
I know, I saw.
yeah, I'm scrolling through, but I don't see any. Any more questions? We still have some little, a few minutes. Well,
my hand is up now. Do you hear me?
Oh, I hear you.
Okay. So this is related to the last question, and what I was thinking of the general overview that I got of your data as you were going through these many things that you've collected was that there just seemed to be a lot more variability within the less urban populations than within the urban populations. And so what I was wondering, how would you distinguish just an environmental filter that maybe not everything that's out there in the environment survives in an urban setting? How would you distinguish that from adaptation to an urban setting?
Okay, yeah, so that kind of brings to mind things like epigenetics or maternal effects. I would say that in the case of our reciprocal transplant experiment, if the results that we saw for that experiment are the result of maternal effects or epigenetics and not local adaptation, the maternal effects and epigenetics are still resulting in plants being more fit at home. I think the best way to kind of start answering some of these questions is to do population genomics to see how urban and rural populations are related, because there's a lot that we don't know about shepherd's purse. First of all, we don't know the primary way that it's vectored. So the seeds of shepherd's purse might be bird dispersed. which is something I'd like to look into. If that's the case, that really opens up a scale issue in terms of looking at this environmental filter that you're talking about. Because if the plants are highly plastic and they're being vectored by birds, it may be that it's just the plasticity will be what allows them to stay or not stay, and not so much, they might be shifting so much, they might have not enough time to even evolve locally. I will say some of the places where I collected seeds, I went to visit the subsequent year and there were no shepherd's purse there that year. Other sites had shepherd's purse again. So we don't know how long a certain site stays, seedbed site stays active with shepherd's purse. We don't know how it's vectored. It might be vectored by birds. It might be vectored by human and dog feet on a sidewalk, because the seeds do secrete a mucus when wet, and they're very, very small and light. And so dog feet and humans walking, or even rat feet and stray cat feet, you know, walking on a wet sidewalk near a plant that just released thousands of seed pods could vector them down the sidewalk. And so we need to do the population genomics so that we can understand, okay, are all of the plant individuals in New York City from a source population, or so how genetically related are all the populations, or are a good subsample of the populations in New York City? Because that might give us a better idea about vectoring. It's a tough question.
Thank you.
Okay. So I think we are out of time now. What is it? Yes. So let's thank her again.
Yay, thank you guys. Oh.
Congratulations. So
Congratulations, Rebecca.
Thank you guys so much. Thank you so much for coming. I really appreciate it. I'm happy it's over. That part's over. That part's over.
now we have to the classroom. There's another class coming. Sorry. I wanted to say that all the time. Okay, so only the committee stays in, please. Bye, everybody.
Thank you, everybody.
Everybody.
So much.
Thank you for coming. Thank you for attending. Such great attendance. Super.
Thank you. Bye, Professor T. Thank you for coming. It's good to see you. You're still muted, Professor T.
Okay, good to see you. Great job.
Thank you so much for coming. I appreciate it.
Talk to you soon. Bye.
All right, we'll talk soon.